Okay, so it's been 4 hours later after I started my restriction digest, and now I'm coming back, and there's been another team that's added some samples here that are going to be necessary for this next step. Now, what we're going to do now is we need to clean our restriction digest. So our next step is going to be setting up a ligation reaction. And of course, ligation is meant to weld our insert DNA, our PCR fragment, with our plasmid vector. Now, the restriction enzymes are designed to cut the PCR fragment in the plasmid. So if we try and ligate the samples while the restriction enzymes are still in there, their competing reactions will ligate it, the restriction enzymes will recut it, religate, recut, religate. So we've got to remove those restriction enzymes out of the way, as well as any small pieces of DNA that were resultant from the cutting of the plasmid and the PCR fragments. So that's why we need this cleaning step in between restriction and ligation, to remove enzymes, small pieces of DNA, and to change the buffers out. So in order to maximize our recovery and concentration, what we're going to do is have you coordinate teams so that you're going to have two partnerships combine their samples. So for example, right here I have team 2-1 and team 2-2, and there's these are the PCR reactions. So my team 1 is going to take, so we'll set the PCR reactions aside, and my team is going to take the 2 PQE31 digest right here, and we're going to take both of these samples. I've already communicated with the other team that we're going to do this. So we're going to take both of these samples and we are going to use them for our cleanup, and then they'll do the PCR reactions, and then later when we go to do the reaction, or the ligation reaction, we will share those two cleanup procedures. And this just allows us to get our samples twice as concentrated because we're starting out with twice the material. So the first thing I'm going to do is combine both of these reactions into one tube. I've got 40 microliters in here. Again, these are both the PQE31 digests. I can throw that tube away. And I'll add my reaction. The next thing I need to do is I'm going to add 400 microliters of a solution called PB buffer. PB buffer is just a buffer that comes from the QIAGEN manufacturer where we get our cleanup columns from. And so I'm going to take 400 microliters of PB buffer. Now I'm going to take one spin column right here, and the spin column itself, so that's the column. This is a collection tube, a 2 milliliter collection tube, and you can see it has this white frit or disc in the bottom of it, and that contains the silica gel. The silica gel has a positive charge on it. Our DNA is negatively charged, and when we wash our sample through that silica gel disc, the DNA is going to bind to the silica gel under the conditions we're using with the PB buffer. And the other things like carbohydrates, proteins, very small pieces of DNA, nucleotides, those are all going to flow right through and into the collection tube. Okay. So what I'm going to do is I'm going to take my pipette and I'm going to load the entire amount, throw that tube away now, into my collection tube. And now I need to spin and centrifuge this through. So I'll open it this way. So in my centrifuge I'm going to load my sample. Now I actually need to balance this a little bit. The tube weighs quite a bit, so I'm going to balance this. An empty tube on the other side. Make sure my lid's on. And then I just need to spin this for about a minute. So I am going to decrease my time to 60 seconds. And I'll do it at maximum speed. Okay, so our centrifuge now is winding down. The centrifugal force placed on that tube has pulled all of our solution down through that membrane into the bottom of the collection tube. So automatically opens there. Pull it off. That was my counterbalance. And so you can see that now I've got that 500 microliters or so down at the bottom of my collection tube. I'm actually going to dispose of that flow through. Don't need it. My DNA is bound to the column up top. Okay, and the next step, I'm going to wash that DNA to get rid of any remnants of protein or that might have been caught in the membrane. To do that, I'm going to add 750 microliters of PE buffer. So PE buffer will be sitting on the counter. It has sort of a tan label on it, buffer PE. Again, this is provided by the manufacturer of the DNA isolation kits we use. That's the PE buffer. I'm going to add it. You want to make sure that you stick your pipette tip inside so you don't get the PE buffer around the ring or the edge of the column. And this time, just to balance it a little bit better, I'm going to go ahead and add actually the same volume of water into the bottom of this tube. I don't want to run it through the tube because I don't want to contaminate the collection column. So I will, that's my, label this one as my sample. Put this on. And we'll centrifuge that for another 60 seconds. Centrifuge popped open, so now I can pull out my samples. That's my sample. That was my weight. So I can go ahead and put this on the side. Alright, now I'm going to discard that PE at the bottom. And one of the problems is that there can still be a little bit of residual amount of PE which contains alcohol in it, and that'll ruin our future reactions. So I'm actually going to do a final centrifugation just to get any remaining PE buffer out of there. So that's another 30 seconds or 60 seconds. Okay, so now I will remove my sample. And you can see there's just a little tiny amount of PE that was still in that column that we pulled out. Now, that's my last flow through, so I really don't need this collection tube anymore. The last step is I need to elute my DNA. My DNA is still bound to that little membrane in there, my silica gel, and so now I want to elute that off. And it elutes off by changing the conditions of the flow through. So now I'm going to add a slightly basic amount of water, so low salt, high pH is going to elute the DNA off of there. My collection tube now changes. This is a Eppendorf 1.5 microcentrifuge tube, and that's what I'm going to collect my sample into. And I'm going to elute with about 40 microliters. In this case, our water is basic enough to do it, so I'm going to just pull out some double distilled water. And on doing this, I want to add, I don't know if you can see my pipette tip, I want to try and look inside and add it just on top of that membrane. I don't want to touch the membrane with my pipette tip. Put it back in my tube. And now I need another sample. So I can take, that's my real sample right here. This was my dummy control, so I'm just going to treat it the same way. Now you'll notice that the lid will not cap or come over the vial, and that's okay, it doesn't have to, but I do have to be careful about the way I set these in, or the lid will snap off, cause some problems. So I want to set them in, and I want to make sure that the lids are opposite each other and that the two centrifuge tubes are opposite of each other. Just going to let that sit for a second before I start it out, so that the DNA has time to go up into the solution of the 40 microliters that I added on that column. So we'll sit and let it wait for about a minute, and then we'll centrifuge again. So it's been about a minute, and I'm going to start my centrifuge. Alright, my elution centrifugation is done now, and as I open this lid, I do want to show you one thing that I did do. This arrow on the centrifuge, it's probably hard for you to see, but this arrow is pointing and telling me that the rotation of spin is that direction. And so as I placed my collection tube and column in here, one of the things that I did was I made sure that my lid was actually trailing. So as this spun, the lid was acting as a tail trailing behind, and that seems to make it not break so often. Alright, so now I can pull out my sample, and then this was my old collection tube. I haven't used that at all, so I'm just going to put it back. And there in my sample tube now, the column, all my DNA is eluted. You can see that's 40 microliters. And what I want you to notice is we originally had 80 microliters that we added to the column, 80 microliters of sample with some PB in it, and now we've eluted off 40 microliters, so we've effectively doubled the concentration of our DNA, and that's why we wanted you to recombine it at the beginning. I don't need my column anymore, but I do need to take my now clean DNA, and I need to label it. So I'm going to label this as 2-1, and this is going to be clean PQE. Now the other group is going to come along and they're going to clean my PCR fragment, and so they would label theirs 2-2 or whatever their group number is, and they would label it clean PCR. My clean PQE31 and my clean PCR are now both digested, and they're ready to go on to the next step, which is going to be setting up our ligation reaction.